Hi, I'm Josh McGinnis, and this is the very first edition of the EverymanBio podcast. If you're giving this podcast a listen, I want to thank you for giving me the opportunity to share my love and passion for all things science, biology, and technology with you. In this episode, I'm interviewing a gentleman by the name of Dakota Hamill. Dakota and I first met over 10 years ago after he responded to an ad that I'd placed on Craigslist seeking used chemistry glassware. After losing touch for a while, Dakota and I recently reconnected, and since then, he has been a supportive figure on my own journey to self-learn genetic engineering and to found a new biotech business. Dakota is a kind person who carries an optimistic view of the world, and I think like so many of us, he wrestles with the realities of being an entrepreneur and a scientist in the current times and how to balance his desire to improve the world by doing good when there's so many forces working against him. Dakota is the co-founder of the biotech startup Prospective Research based in the Boston area. He's participated in biotech accelerator programs across the world, and he got his start by building a fully functional DIYBio lab in his basement where he worked on antibiotic discovery. Dakota has almost 2,000 subscribers on his YouTube channel Basement Biotech, so be sure to go and check him out. I want to thank Dakota for his time, and I hope you all enjoy listening in on our chat. We covered what I think are some pretty fun topics related to education, ethics, startups, biotech, and so much more. Enjoy.
Dakota, thank you so much for doing this. You're like the first podcast guest that I've had so far, so it's awesome.
Nice.
And it looks like you're in your lab, which is amazing. So I guess let's just start with telling everybody like who you are, how did you get started, where are you at now? Like you look like you're in a lab setting.
Oh, it's quite a long story. Yes, we are in the lab of a startup I started with my friend 5 years ago, actually, back in 2015. It's called Prospective Research, and we focus on aquaculture microbiome therapeutics, effectively using bacteria from the environment to try to replace the use of antibiotics in medicated feed and managing disease. And it's been a wild ride here. We started in a lab in my parents' basement we built with some stuff off of eBay. We had $2,000 to our names. We didn't get into the graduate schools we wanted. And after we became friends in college, just started this giant crazy adventure. And I mean, I could ramble on for quite a long time into how we got started and where we came from, but.
So you're at a startup you started now.
Yeah.
Wow. So and how long have you been going at this startup?
It's April 2015, actually. So a friend and I graduated from college with just bachelor's in chemistry degrees in 2012. Each worked for, he worked for a bigger biotech company. I worked for another startup for a year and a half. We applied to grad school, didn't get into the schools we wanted to, and said, you know, we saw some posts on the DIYBio forum actually of this new accelerator called DIYBio, and that was going to be the second batch of synthetic biology biotech accelerator following in the steps of Y Combinator. And we managed to apply to the cohort 2015 in Ireland and got in there. So that's where we raised our first 100 grand and dropped everything and moved to Ireland for 3 months. And the rest has been a lot of ups and downs, moving back to the basement, then to a small incubator at a local college, and then finally into our own building last year.
Wow. That's quite a journey, and I have tons of questions I could ask you about the entrepreneurial journey you've been on. But it sounds like you started. Would you say you sort of started the DIYBio stuff like right out of high school?
I'd say I interned at, kind of out of college. DIYBio wasn't even around when I was in high school.
Yeah, that's what I was going to get at. It was like, how did you? What? Yeah, it seems like a relatively recent phenomenon. Like I want to say 2009 was really when I noticed that it exploded. But how did you find out about it, or did you, or were you just kind of like you intuited it? You just kind of like, I want to do something on my own. I'm just going to start buying some stuff. Like, how did you even find out about it?
I mean, in high school, I was tinkering around and ended up interning at doing bio labs, and I learned more in that summer, just interning at doing bio labs on DNA repair mechanisms and enzyme mixtures than I did in probably 4 years of undergraduate school, because jumping into hands-on skills is something that teaches you much more than just memorizing stuff for worksheets and homework. But after that, I just always loved science. I didn't finish high school. I ended up dropping out and cutting trees and doing manual labor, but always loved science. And when I went back to school, like 2007, 2008, ended up somehow, I guess through Google terms, finding Matt Cowell and Jason Pogue, who were the original DIYBio founders and are still around. And went in one day finally to meet Matt Cowell at Boss Labs. Actually did meet Jason, I think, at a Genes to Genomes conference he used to have. But I mean, this is over 10 years ago. So, yeah, just random Google, and then coming into Somerville, Cambridge, and finding the people that actually started the Google group, and from there networking with a bunch of really interesting people all over the world over the past couple of years.
That is awesome. But I want to get back to something. You said you didn't graduate high school?
Did not graduate high school. I dropped
out. My first thought when I hear that is awesome, because look how far you've come. Like, and everything I'm doing with EverymanBio is sort of like trying to demonstrate that you can self-learn science. You can teach yourself a lot, especially today, because we have so much access to information through YouTube and books that I bought for really cheap. And here you are after your ups and downs, but you're still, look where you're at. You're in a legit lab. You've got your own startup. You've raised money. You know, I'm sure there's lots more that you want to do and where you want to go, but you're sort of a great, you know, story, I think, for what's possible if you stay focused on a goal and really apply yourself to something that you're passionate about pursuing.
Oh, without a doubt, if you want to do something, I firmly believe that the universe will help you do it. And that's after many dark nights, but many interactions with people that the way that they happen just seems so, you couldn't have written it in a book how we ran into some of our investors and the help that we got from people. And yeah, not finishing high school was in a way depressing, but I had always loved doing science in school and after school, and the hands-on stuff like you're making videos about and teaching now, that's what really gets me excited. It wasn't doing the busy work. And unfortunately, if I could go back, let's just say that there are some hoops that if you jump through, it makes your life a lot easier. Like jumping through the hoops of finishing high school and going to college. But in a weird way, carving your own path makes you just, I don't know, hungrier to achieve what you want to achieve. And the suffering pays off eventually. Hopefully. It'll make a good story if it all works out. Otherwise.
Well, 100%. There's definitely something to be said about that. And don't get me wrong, I'm not advocating there's not a place for education, because I absolutely think there is, especially in the STEM fields. But I don't think that's the only path, you know, and I think like so many people grow up being ingrained with this idea of there's this one way to do things and one way to be successful in your career and as an adult. And it's just not true. You know, and I understand why people push that model, because it worked for a lot of people for a long time. And as a parent, you have fear. You want your child to be successful and to do well in life. So it's a little bit, you know, probably there's some fear with leaving it unknown and leaving it open for the child to kind of explore, you know, their own way and their own path forward. But I'm very much like you. I mean, there's times where I like to go really deep in a particular topic, but the best way for me to learn is just to sort of definitely be hands-on, but absorb everything, read a little bit, do some stuff, get stuck and have to problem solve. Because when you get stuck on a problem and you have to go research the solution, I find like that's when I accumulate all of this knowledge. And I picked that up in the software engineering world because I'm also self-taught in that. You know, it's kind of been my career up until now. So
without a doubt, I think that's one of the fundamental flaws of science education is, let's say, I'd argue for math that there are particular ways that you need to solve mathematical equations, and there is a right way, so to speak. But when it comes to the sciences, especially in school, even in college, you're giving this checklist, this cooking list of do X, Y, Z, and if your solution turns blue, you get an A. If it turns red, you did it wrong, and you got to do it over again. So there's none of that open-ended exploration, which is what science fundamentally is, trying to use the skills and knowledge you have to find out something new and unknown. And when there is a right or wrong answer in a lot of the ways that schools teach it, there's no thinking involved, no critical thinking, no exploration, and that's why it gets boring. But that's why DIYBio is so cool, because it's people that want to learn stuff. They don't know what they're doing, and in some cases come together, and there's people on the forum that are great with plant biology or plasmid design or gene editing, and there's other people that are experts in electronic engineering. So that was the best thing, is finding a group of like-minded people that just wanted to learn and maybe didn't fit into the academic niche or the business niche. And I'd say we're all a little bit of outcasts, but in a good way.
Yeah, I think we need that. I think we need as many people as we can that come from different backgrounds, varied backgrounds, thinking about problems in different ways, all banging on the same problem set, because then we're going to get these like interesting creative solutions that we wouldn't have otherwise expected. Yeah. And I also think, like, on your topic of the critical thinking, I absolutely agree with you. I just have opinions in general about just the whole education system not teaching people how to think critically and how to appreciate nuance and context and pick apart things and really look at them under fine detail and consider all of the possible, things that could be going on with whatever it is that you're observing. So my own experience with that is that as a self-taught DIYBio guy, like today I did my first PCR. Like that for me is a big goal of something I've been preparing for a long time. And it's actually sitting over there. And now when we get done, I'm going to be running my first electrophoresis. I'm super excited about that. I don't expect it to be successful the first time around, but that's okay, because I'm already so much further than I was last night when I did my first try. So I think in another week of banging at it, I'll probably have figured it out. But what I discovered was, even when you're reading through protocols, like I'm following a protocol, which is kind of like a recipe for performing a procedure. It's like how to bake a cake. Here are the ingredients, here are the tools you need. But when in the real world, you don't always have the same ingredients. You don't always have the same tools. You definitely don't have the same skill sets or the same environmental factors. So as I'm going through the protocol, I'm beginning to see that there are elements about this that I'm going to have to adjust for my unique situation and scenario. And that's the challenging part, because otherwise it would just be like baking a cake using someone else's recipe. But there's little trade-offs and little minor adjustments that you have to make to it. And I think that's where the problem solving part of it comes into play. That's where the hands-on practical part of it comes into play. And maybe something that isn't as taught in traditional education systems. I don't know, I can't speak for that, but that's kind of my thoughts on it.
I remember your story reminded me of a story when I was like a sophomore or something. I was really looking forward to taking organic chemistry too, because there was a German professor that was teaching it, and he was known to be a very good chemist and very hard, or I'd heard that he was very hard. And come to find out, he had ended up getting fired the year that I was going to start with him. And I kind of asked some of my professors that I were friends with, like, why did he get fired? And students were complaining because he was too difficult. And the example story he gave me, or they gave me, was that they needed to do an organic synthesis reaction, and it had 2 reagents. So you needed to mix the reagents together in a solvent, and out comes your product. And the professor asked the students to choose their own organic solvent. Did you want to run this in DCM or ether or ethyl acetate or heptanes? Pretty simple, nothing crazy. And if you looked at the structure and the polarity of your compounds, or maybe just tested the solubility, you might be able to have an educated guess on what solvent to use. And I mean, that's what you'd have to do in the real world. If you were doing anything new, you'd have to use the skills you got and said, what's the solubility? This is the best solvent to use for that reaction. And so instead of just telling the students, add this and that to that solvent, stir it for an hour, take a melting point temperature and run a TLC and you're fine. He got fired because he made the students think. And I was just like, wow. So that was basically when I started messing around in the lab after hours, because I went to a small state school and no PhDs are offered, no masters, so nobody was really doing research. So it was cool because my friend and I got free reign in the lab. We got to use the NMR, the LCMS, because there was nobody competing for it on time. So that was pretty cool to have free reign in the lab and learn, but
that is
kind of
awesome.
sucked not having a mentor either.
Yeah, yeah, and I experienced that too. I mean, there's trade-offs with all of these different approaches, and I'm certainly not advocating that someone, um, abandon any kind of mentorship or like. I've spent so much time doing things and trying to learn on my own, but things would be much faster if I had a teacher here that was like,
100%.
no, 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 you don't push the pipe all the way down. There's actually a stop beforehand.
Yeah, first resistance.
Yeah, so that, like little things like that. So definitely there's a place for mentorships and formal education, but I just also want to advocate for the self-learning, self-taught, hands-on approach as well. There's definitely more of that I think we could do, not just, by the way, in biology. I just think in all the trades in general, across the board, especially as like our economy gets more specialized, and the whole other area of conversation that we could go into. I did want to ask you though, when you first started with your DIYBio stuff, was there a project, like I'm starting with fungal DNA barcoding because that's giving me the opportunity to practice and learn all of these skills that will take me into the genetic engineering side. Was there something that drew you to it that you were like, one of the first things I want to do in DIYBio is accomplish or do this project?
Yeah, that's a good question. I'd say there was two things. I mean, when I was really first starting on the DIYBio, I remember getting excited about the type of excitement that I'd find from very simple things. I'd go on the DIYBio forum when I was in college. I was 22 or 23 and asked. So I'd eaten a bunch of celery one day and my tongue was going numb. I was like, why is eating celery make my tongue go numb? And then somebody on the forum said, oh, it makes eugenol, and eugenol is actually a local anesthetic. And then I look it up and, wow, eugenol is actually used to knock fish out as an anesthetic in water. And so reading into like Wikipedia, getting lost on chemical compounds and natural products. But the one night that I'd say really changed my direction in life was because I wanted to be an organic chemist, but then organic chemistry seemed to die back in the 40s or 50s or 60s after World War II, and now it's a lot of biology. I was up at like 2 a.m. one night because I couldn't sleep, and I went on PBS, and there was it wasn't Nova, but it was a science documentary. It was called Jewels of the Jungle, and you can find it online. And it was the story of Gary Stropel, the scientist from Montana who studied endophytes, these symbiotic, some don't call them symbiotic, but however they get into the plant tissue, they're fungi that generally live within the vascular tissue of plants. And so immediately became obsessed with endophytes and fungi and really started culturing, we were sterilizing the outside of twigs and leaves, and it was amazing to actually see the mycelia make their way out of the vascular tissue of plant material and onto your PDA petri dish, and just this whole wonderful world of fungi. You'd say, I thought fungi were mushrooms that grew on the floor. I didn't know they lived inside of plants. And so he went on this whole journey. It follows this whole life's research of how the first billion-dollar anti-cancer drug, Taxol, was found in the bark of the Pacific yew tree. I think I've said this story before, but then Gary Stropel and maybe some other scientists found an endophytic fungi that produced the same compound. So there was questions of exchange of DNA over quite a long time, probably by evolution. And it made perfect sense. And that's what really got me into natural products. And yeah, it started with just culturing endophytes, doing a little DNA fungal barcoding, and then we got into DNA barcoding fish because I'm from Gloucester and it's an old fishing town. And from there it jumped to iGEM and synthetic biology, but we were never at a school that was going to sponsor an iGEM team. So it kind of went back to natural products. I said, if our school's not rich enough to have the tools necessary to do synthetic biology, I'll just rely on nature to do the biology for me and try to be a bioprospector. So that's the history of the company, is bioprospecting. It's looking to nature and the microbes that live bottom of the ocean, your backyard, the oak tree in your backyard, and seeing what amazing compounds they produce.
That's amazing. So like synthetic biology, that seems to be a relatively new field. I know it's sort of been around and been obviously been worked on in various facets over the last decade or so. It just seems like what's happened I'll speak from my experience as a software engineer, is now's one of the best times to self-learn and develop a career in software engineering, in part because it's become so abstracted that people that don't have to have this really deep mathematical, traditional computer science background in order to be a productive software engineer that's developing applications for companies. And that has a lot of value associated with it. It seems to me that synthetic biology is headed in that same direction. When I am researching, designing plasmids to modify yeast, for example, it seems like we're getting closer and closer to being able to take genes that have already been identified and understood and sort of look at them as libraries that you can plug into the plasmid. For me, as a software engineer, that's great. You know, there's so much software that we use where we're using abstractions on top of each other. We're not having to dig in underneath the hood. And, you know, if let's just use an analogy, if we want to build a house, we can just grab the wall, we can grab some nails and a hammer. We don't actually have to worry about building the hammer or finding the materials to put the wall, to actually make the wall. We can just get that component and plug it in. It seems like synthetic biology is heading in that direction as well, where there's these levels of abstraction that are allowing it to become more accessible to non-super scientific people who have PhDs and have gone, like, who can do the really deep. And I think there's a place for all of them. Is that true? I mean, is my instinct right there? And to me, that seems incredibly powerful, because it means that synthetic biology will become much more broadly accessible to, like, intellectually inclined people that are interested in it, like myself, but also a little scary, because just like in software, yeah, you can glue all these things together, but if you really don't know what you're doing, you'll build really slow software, or it'll have security issues. And I presume the same thing would happen in synthetic biology.
Yeah, that's a very multifaceted, deep question and I haven't spent a lot of time, almost no time, in the synthetic biology field besides following it with great interest in Ginkgo's work and the iGEM competition. That's about as close to synthetic biology as I've gotten. But the fundamentals make sense in that people have spent the past 30, 40 years learning how to program computer code to translate into zeros and ones to feed into processors. And now the code of the future is ATCG. How is the DNA in any organism put together to make an unlimited number of molecules, proteins that have all these amazing functions? And people that are really pushing the boundary or understanding or trying to build the tools that do allow you to sort of plug and play. I mean, it sounds easy to say, we have a protein that encodes GFP. Let's take it out of jellyfish and put it into a bacteria. And now it's super easy. You can buy a plasmid for $5 or $10 and do that yourself. I imagine the people that did that and won the Nobel Prize, I don't know how long that took, but I imagine it took quite a long time to do that. So that speaks to how farther along the science is coming. I think iGEM is a very neat idea when the people who founded Ginkgo, and I'm sure some other scientists, said, how do we standardize synthetic biology for the future so that when somebody in 20 years wants to go build something, the guy or girl that originally designed it 20 years ago, who used a different hammer and a different screw, can pick it up and basically use the same set of tools to work on it and develop it. And outside of a couple companies, and I'm sure a lot of institutions, I imagine over time things will become more available to the public. Like right now, you can get these basic kits for, I wouldn't even call it synthetic biology, it's just transformation, traditional molecular biology. But in the future, it may be that you go and you say, you look up compound A and you say, I want this DNA sequence of that optimized so that I can express it in yeast. Because it's only a few expression organisms that people use. There's yeast, E. coli, maybe some bacillus. I'm sure there's some others, but it's not like there's tens of thousands of novel species that are being used to express these recombinant proteins. So there's still a lot to build, and it's so complex. We're not at the stage yet where you can go to an organism, find its gene, and say, I want that gene sequence in that organism, and I'm guaranteed 100% to get the molecule I wanted, the protein I wanted.
That makes sense. Yeah. Yeah, there's Yes, yeah, definitely. There's definitely other factors involved, like making sure that the gene gets activated. But even things like synthesizing compounds often involve multiple genes working in tandem, and there's sort of a pathway involved in a handoff between one gene to the next. And there's certain factors that are involved in that handoff that need to be, the environment needs to be correct for those things to happen.
Yeah, intermediate pathways and shunt pathways, and everything needs to come together. And perhaps it was built perfectly in the natural organism, but moving it into another organism can spell disaster. I mean, people have spent hundreds of millions of dollars trying to do that, say in the field of drug discovery, and in some cases aren't successful because nature's designed this stuff for a very long time, and it's amazing, but it doesn't always work once humans get a hold of it.
Yeah, definitely. That makes me think, like, something I've been trying to figure out and think about is like ethics in DIYBio, and where are the lines, and what's good citizen practice for, like, what should we be advocating for in terms of the general public, and both in terms of what we should do and educating the public, because there seems to be this line where I think I kind of reminded of the Anarchist Cookbook back in the day. I don't know if you remember that.
I know that book, yeah.
It was like banned and people tried to keep it hidden, and it had all of these recipes for how to build bombs and all these sorts of things. But it was an interesting test of free speech and knowledge. And so I wrestle with this with DIYBio, or just even synthetic biology, especially when you talk about modified organisms. There's the potential for a lot of danger there. And it is getting easier, even though it's not completely 100% accessible yet. The pace of advancement is accelerating for sure. And so there's a lot of power there. And part of me feels like we need to make known how it works and what can be done to as many people as possible, so that we can, at the very least, we can appoint the smart people, the right smart people, to look after us, to make those decisions on our behalf, the people that actually know how to put it together. On the other hand, if you reveal too much information and you make it too easy, I mean, it's inevitable that the technology is going to get to the point where we could just build, there's going to be a website that lets you have a list of components and you can drag them over, design the gene expression that you want and the organism you want it to go into, and have it delivered to you. I mean, we're really, really close to that already. That's dangerous, right? I mean, that's not something I think we want to have in every person's hands, because inevitably we will have a bioterrorist that wants to take out, his city or something like that. So have you thought about that at all? It's something I'm trying to wrestle with, and I haven't really figured out those philosophical boundaries yet for where the line should be drawn and how to establish them.
Those are really good questions. I remember early on, I think a lot of people, or at least myself, I always said, I wish there was sort of a Hippocratic oath for DIYBio people, like first do no harm, and then a whole other list. When I was starting, I was always aware to not do experiments that a home lab wasn't set up for. Don't work with crazy flammable solvents or toxic chemicals. When it comes to biology, use organisms that are BSL-1 or safe, or at least take the proper precautions, because not just yourself you're putting at risk, it's whoever lives in your house or your neighbors. So I think a lot of people that started out were very self-aware and conscious of, look, if we're starting this new movement of allowing people to start doing science at home, it's not the same thing as ordering some resistors and transistors and doing soldering at home. There could be implications. And so sort of anything that we do and we do publicly, which was on the forum group, and some people have gone beyond that, and I'd say done some theatrical stunts to draw attention, we kind of set the tone where all it takes is a couple bad apples to do something too crazy, where all of a sudden society says, these people should not be having access to this stuff. And then it hurts the people that really want to do science. And you know, there was an example. We used to go into Boss Lab in Somerville. There was an FBI agent that used to come there. He was very vocal about who he was and why he was there. And in the early days of DIYBio, they basically came to check out, you know, who are the types of people that are doing this? What projects are they working on? Okay, you know, they're trying to get glow-in-the-dark Vibrio from shrimp, or they're trying to do some PCR. They're not trying to hurt anybody. But I do remember one time going to the conference in Harvard, and there was a kid that had been hanging around, and you know, we were kind of throwing ideas around about what we wanted to work on. And he's like, when can we start making viruses and putting them on door handles? And so we kind of, as a group, said you're no longer welcome, and sort of self-regulated that. But there's literally nothing stopping people from doing bad things. And I think the best any normal person can do is just be responsible. And as a community, I think there are some websites out there where we've tried to set a list of, you know, these are sort of the code of ethics to not draw bad attention to the movement and be responsible. When it comes to the ethics of synthetic biology on humans and some of the crazy stories, I don't know. That's why I kind of like working with natural bacteria, because I don't have to think about those things.
Yeah, I've seen a couple of those folks. I know there's some people on YouTube that have designed some things, some vaccines, and are injecting themselves with GMO things.
Yeah.
And you know, on the one hand, I've said this before on other live streams, I definitely think people should have autonomy over their bodies. But I think that when you do something potentially to your body that could end up escaping outside your body and end up hurting the general public, then it's maybe crossing a line there.
Yeah.
And, you know, I'm generally like not a big regulations guy in general, because I think it can slow down innovation
100%.
in science. However, as I get older, I get more wise, and I start to see the trade-offs with things actually moving too quickly. I mean, even in our society, as technology becomes so prevalent and the skills required to have a job are so specialized, we begin to abandon and leave out large swaths of the population who will never be equipped to do these highly specialized jobs. So how do we deal with that? And on the one hand, you could argue, well, maybe we need policy that kind of slows and puts the brakes down on some of the technology. On the other hand, there are competing countries that have no objections to any of that. And there's danger in allowing them to run away with technology uncontested, and for us, you know, here in the US to maybe fall behind with that. So there are these competing things that I'm still trying to wrestle with in my own mind about, like, what should that underlying philosophy be, so that, one, I can advocate for myself, but also like share that with the world. It's just something I like to think about from time to time.
Yeah, that is a deep question, and one I don't even think I'd attempt to answer. I think I like to believe in the goodness of human beings, and that we're all here trying to better the world and do fun science to help people, or just out of curiosity. But as you said, as I've grown older, I've gotten a little wiser, and I understand that there are forces in the world that don't share that desire to always do well and do good. And so in that instance, some of this Department of Defense research and funding, I mean, it has a role. You can't ignore that human beings do have a long history of wanting to kill each other, unfortunately.
Yeah, unfortunately, so. Yeah. Maybe we'll find the gene for that one and knock it out. Or yeah, maybe there's a role for it. Maybe Mother Nature created it for a reason.
Yeah.
Yeah. Okay. So switching gears for a little bit, I'm getting ready to put together a pitch deck and approach some investors about my little biotech company idea. And so I've been in a totally different industry. So I don't have a lot of industry experience in biotech and what it's like with starting a company and raising money and things of that nature. What advice would you give to someone who wants to have a startup, maybe go raise money? What are some of the tough lessons that you've learned on your journey that maybe you could share with me and anybody else who might be interested?
Oh man, tough lessons. Well, one of the first VCs that taught us how to pitch, he ripped everybody apart. And just long story is that, tell a good story. because human beings have used storytelling for a very long time to pass on useful information. And as much as I love science data, and as much as I love talking sometimes and pitching, you know, when you read a really technically heavy deck, sometimes your eyes want to fall asleep. And you know, human beings, as logical as some people are, we're still very driven by emotions. And so when people get passionate and excited about stuff, which I tend to always do because I just love what I'm talking about, that passion comes off and somebody else gets excited simply because you're excited, not because they care about secondary metabolites from actinomyces. They probably don't even know what they are. They're like, I don't know what this is, but this kid's really, really getting me excited about these terms I don't understand. So passion's one big thing, and storytelling is another, using pictures and driving these points across. But I think if I could do it all over again, I would not, this is a hard one. I don't want to get in trouble for saying this. I would build a minimum viable product and try to sell it before I ever tried to raise money for a company. Because a company that makes revenue on day one or has people that have validated their product idea, that's very good. I mean, we've spent years and not sold anything, because I mean, that happens a lot in biotech. You work for decades sometimes to get one drug through, or decades to get one platform technology built. And oh man, it'd be real nice to have a product that's just validated early on, you know, and maybe not friends and family. Now with the internet, you can literally get strangers you don't know who aren't going to be biased and say, yes, I want that. Yep, that solves the problem. Nah, I wouldn't buy that. And they might even give you feedback.
That's actually just generally good advice for starting any business.
Yeah.
Try to get it as far along as you can on your own, because it gives you leverage when you go to the investment community. If you can
100%.
go to an investor and say, look, I already proved some demand here. I've already proved the technology works. I just want to scale. Well, that's an exciting story to an investor, because they'll give you money all day long if the assembly line is going to print more money at the end of the day. But when you go to them and say, like, I have this idea, I don't know if it's going to work. I don't know if there's demand for it, but can you please give me $1 million so we can go on that journey together? You're going to lose a lot of control over that company. You're going to lose equity in that company. And there's trade-offs with it. It's not to say there's not a place for it. And I think one of the things that I would say is maybe different than the tech world is biology seems to have more of those unknown components involved with it, especially when it comes to like engineering organisms as an example. Versus software. I think the software world and tech world has just iterated and evolved so much in the last 10 years that there's a lot of tools and techniques you can use to quickly build a proof of concept and test if there's demand and if it's going to resonate with the market. That's not necessarily the case. My gut tells me with a lot of things in biotech, because a lot of it just doesn't exist, and a lot of it requires a lot of R&D to get going and get built. So yeah, I think that's good advice. I feel your pain. I mean, anyone that has to raise money is going to somewhat be beholden to their investors in a lot of their requirements, and that's not easy. For even publicly traded companies are beholden to their shareholders, not necessarily to what they want to do within that company, but to constantly keep, becoming profitable and increasing the market share. So every company at every stage seems to have to deal with that sort of trade-off. But I think you're absolutely right. Try to do as much of it as you can in the beginning, and then you have some leverage when you go to the table to these investors.
Yeah, and also nowadays, I'd say that accelerators were some of the best experiences we ever had. To be able to take an idea, and a lot of accelerators willingly welcome ideas that could have big payoffs if they work, but they're perfectly okay with sometimes not even having a proof of concept. But accelerators that give you, a small amount of money, not small by any means, $50,000 to $250,000, sometimes less, sometimes more, and 3 to 6 months in a concentrated space surrounded by people that are trying to do the same thing with a lot of mentors. We've gone through 2 accelerators now, and they've both been the most intense, fulfilling 3 months of our work, because you just thrown, in our case, across the Atlantic Ocean into Europe, and really surrounded by people that are in the industry you're trying to get a hold of all the time. I mean, we're north of Boston, Massachusetts, and biotech is big here. But now that we're in the aquaculture space, it's not a lot of aquaculture going on in the United States. So it is important to think about what industry is around you and where do you actually physically want to start a company. There's so many things.
How do you
accelerators are good.
So accelerators, are there biotech specific accelerators? Like is that something you recommend? Like find an accelerator that focuses on biotech specifically if you're going to go that path.
Yeah, I think there is a list now, but SOSV, Toronto Sullivan Ventures, or SOSV, DIYBio, and RebelBio. We went to RebelBio. They are very biotech specific. Y Combinator is very well known because it's a lot of the big, famous California VCs. And they take software and tech companies as well as I think some bio companies. But there's a lot in the Midwest too. And living on the coast my entire life, I'm starting to get a little jaded with the prices of just doing business in Massachusetts. And there's some amazing accelerators in the Midwest, some of them founded by the big agriculture companies out there. And your dollar can stretch a lot further out there. So yeah, there's agriculture specific, aquaculture specific, probably human cell biology specific, synthetic biology specific. There's tons of accelerators these days. That's a good way to get your feet wet. Off the ground.
What's the best way to approach those folks? And so I could ask you a bunch of questions, but I won't go too deep on the accelerator path. But if someone wants to approach an accelerator, what's the best way to put their best foot forward there? And is the entrepreneur expected to like move to a particular location? Like I'm in LA, right? I'm a little bit more established later. Yeah. I'm not that old, but I'm more established in my life. I'm not in a position where I could just pick up and move to San Francisco to do the Y Combinator accelerator. Is that a requirement of accelerators?
Question, because in some cases, yes, they do require the whole team to move there on site for a couple months. In some cases, they only require one person to be there to represent the company. We've seen both. We've heard of companies not getting accepted because the whole team wasn't willing to move there. Every single one we've ever been through will never accept a single person company because their bottom line is, if you can't get one other person to believe in this, why should we believe in you? I don't know if I entirely agree with that, but yeah, I get the philosophy behind it.
Peter Thiel's fund is the opposite of that. They only want solo founders, and they only want college dropouts too. So
it's everybody's got a different spin on it, but in some cases, especially with everything that's going on now in the world, some people are building virtual accelerators. But I'd say the one thing that really stood out the first time we got an accelerator, I'd say the only reason I even found out about it, and maybe that we got in, was because of talking to Cahal, a member of the DIYBio forum, who's been on there for years. He ended up being selected to run their program for a couple of years. And so back to my number one lesson, which is be kind to people, try to help as many people as you can, and just be a decent person that people want to talk to, because it was that network of talking to this person over years, just about random stuff on the internet in regards to science. And it just so happened that when he became program director, I was exactly in the position where I didn't go to grad school. I quit my job. I wanted to found a company. And so it worked. I wasn't scheming 4 years ago, like, oh, if I get this guy on my side, maybe he's going to accept me into a program someday. It just happened that, the universe has a weird way of working out. So it was that network that I think he had the rapport, so that when we had the conversation online, it was like talking to an old friend. And I mean, we did our work with pitching and all that stuff, but
you had already built a relationship with them.
Yeah, I had never spoken to him before, besides over email on the DIYBio threads. And I ended up running into a bunch of people who I now consider friends, who had been on the DIYBio forum, and actually one or two of them ended up being in cohort companies that same year with IndieBio, which is now RebelBio. So a lot of people on the DIYBio forum have gone on to start their own companies, and it was cool to meet them in person and work alongside them.
That is awesome. And on the note of partnerships, I've learned a lot of lessons the hard way about finding people to partner with on your business. Definitely make sure you spend some time building a relationship with that person who's going to be your partner outside of the context of the business. That's my number one thing with partners, because when you go into business, the world changes. And you guys are going to be going through a lot of different things together, having to make a lot of tough decisions. And it's so much more likely you'll be successful if you have something, a deeper connection. It's not just the business itself. It can ruin relationships. So it's really best to have someone that you trust, that you know, and you've kind of established some rapport with. And that takes time. You know, it's like, you can't just decide, I need a co-founder, I'm going to go find them. I mean, you can. Most of the time that doesn't work out, because you're going to basically be hiring your co-founder or your partner, but you're not going to really have developed any meaningful relationship with that person.
Yeah, that's the hardest thing, is finding good co-founders, good team that sometimes are in the right place at the right time, both in the stage of life where you can commit everything to this company, because that commitment and sacrifice is huge. And I ended up founding the company with my best friend from college, who's now one of my best friends in life. And, you know, we've had some ups and downs, and a lot of people said, never start a business with your family and friends. And, you know, some days you understand why, but I'd say the core rock of knowing that somebody's so loyal to you as a friend first, and will pick you up when you're down, and all that stuff, I'd say it has worked out in our particular case. And it's critical that, that emotional business friendship relationship, it can work in your favor, it can work against you. It's like you're married to somebody, but you get none of the benefits. You're around them all the time. You got to make tough decisions surrounding money and both of your futures and everything. Yeah, that's a hard one.
It is tough. And like you said, a lot of factors have to come together for even just forming friendships, especially as you get older. There's a lot of obligations and things that can come in the way of making that connection and bonding. And it just takes time to cultivate it. And every relationship is going to have its areas of conflict. That's the nature of two, you know, sentient beings working together. It's I have a very good friend that always says it's the rupture and repair in a relationship that makes it stronger. It's the repair of those ruptures that actually builds the relationship.
Yeah.
So it's like when you go through something with someone, like that's when you really get to know who they are and bond closer as a result of that.
Definitely. That open communication is key. We've had many ruptures, but the repairs always work when you're actually open and honest about, you know, what pissed you off or whatever. Okay. Now I'm actually better in my other relationships with family, friends, everything, just from the business relationship, because you need to learn how to work through those things without acting like an immature child that protests and says, I'm leaving or whatever.
Yeah, I definitely have had my share of that too. You know, it's just we're constantly evolving and growing. So before I let you go, would you be so kind as to give us a tour of your awesome lab? Because I have never been into a proper lab, and one day I'm going to get a chance to. Maybe I'll fly to Boston and come visit you.
Oh yeah, you're always welcome. We ended up when we were looking for a space, we moved out of the incubator, we wanted a semi-industrial looking space. And so we actually found I'll try to walk around and talk. We found an old industrial space that is one giant open, sunny, old machine shop actually, that we turned into a laboratory. And having this power here has actually been pretty key, but most of the equipment, the autoclaves and the HPLC prep, we get a lot of stuff used. And even some of the companies that our advisors are from now, which are worth a lot more money than us, they started off buying used equipment from eBay or from lab resellers. And I'd say for anybody that wants to start a lab, you know, you can get a lot of good stuff on eBay. So
did you play any role in finding this location and doing things like negotiating a lease, for example?
Oh, yeah.
Can I ask, what does a lab space like that go for? Like, what is it? You're in the Boston area.
Well, this is why we came here 100%. It's a little over $1,600 per month for 3,500 square feet.
That's
and I'd
incredible. Incredible.
Yeah,
you can't even get an apartment for that much for most of Boston.
no. So when we looked at some of the other lab spaces and incubators, I mean, in Cambridge, it was maybe going to be, it would probably be upwards of $10,000 a month. And even in some of the lab space around here, $5,000 it's a month. It's huge overhead. So I still say we run like a startup, even though we're a little bit ways along. We still, want to buy used equipment and save our money. And it's a fine balance between when you start a business, especially in biotech, because there's so much time and research, investors do want you to spend money to get results faster. So it's a balance between spending wisely to get the results you need for your next milestones and also ensuring that you don't run out of money. There's no perfect science to it. I mean, we've had many sleepless nights of trying to figure out the answers to a lot of these questions. And that's why, like you said in the beginning, having a mentor is key in developing that mentor relationship early on. Your network it can save you a lot of trouble.
Yeah, I hear you on that and definitely agree. Questions about your lab. So is it filtered air? Like, do you have HEPA filters up in the air? And yeah,
we have certain circulation parameters to meet for the quality of air that comes in, and most of the work's done in the biosafety cabinets over there. And
so what level of lab is this in a
BSL2.
And is that a pretty like run of the mill, like is that can you do a lot of biotech research at a BSL level 2 lab?
That's really all you need. That's all we'd ever feel comfortable doing. I mean, a lot of college microbiology labs are BSL2. It just means you can work with some general opportunistic pathogens as long as you wear the proper safety precautions, stuff like that. We don't use anything crazy here. We don't even use human pathogens. We're in the aquaculture fish space now.
What's your favorite piece of equipment in the lab? Can you show it to us?
Favorite piece of equipment? Oh man. I would want to say that I know exactly what it is, but I don't. I mean, our new bioreactors are pretty cool. My entire life, I've always said, if I could just get paid to ferment a microbe that produces something, I would be so happy. Because it's like producing beer. You ferment
I was going to say, you're making beer in there, aren't you?
There's your product. It's right there. Actually, these ones just got finished, so they're a little dirty looking.
Oh,
but
sweet. So what do you have in that? Can you actually just take a moment? It's my first time to get a tour of a bioreactor. Can you just point out what are the components of a bioreactor? Because I think someone would see this and go, oh, that's a really complicated piece of machinery. But when you dig into it, it's not so bad, right?
No, and when people talk about rebuilding labware or hardware for DIY bioscience, I think this is probably one of the more complex ones, but a great one to have. It's effectively a glass vessel that you can sterilize, and it has ports for adding in acid and base to control your pH, and an additional spot for anti-foam, and an impeller to control aeration, and an extra line for oxygen if you want it. So effectively, it allows you to grow and carefully control the growth parameters that allow whatever microbe you put in there, whether it's a fungi or bacteria, to grow as happily as it can. So in this, we're growing some bacteria right now, and we spend a lot of time optimizing growth parameters to try to figure out which carbon source does it like, which nitrogen source does it like, does it need extra vitamins or metal salts? And you can track that in one of two ways, just the optical density, seeing more cells means that it likes what it's growing in. And oftentimes, but not all the time, more cells means more of your product because there's more cells making whatever you want it to be making in the first place. I'd say these are kind of, obviously as soon as we got them, I watched my business partner and the other guy that works with us run those most of the time. I don't do tons of lab work anymore, which is a little sad.
What do you do? Yeah, what's your day-to-day look like?
Just endlessly looking through slide decks and sending emails and trying to schedule.
So the business side.
Yeah, all sorts of work.
Yeah.
But it's pretty standard. I mean, we have bioreactors, HPLC, thermocycler.
Are you guys big enough to have, so it's you and your partner, do you have employees? Do you have other people working there?
Yeah, we made our first hire.
Oh, wow, congrats.
Thanks, 6 months ago. And we generally have interns. They just left last week to go back to school. And
Okay.
we do want to grow. It's a fine balance right now of what we need and also the right person, because yeah, when you're at a small stage, the people you bring on are hugely important. You got to balance your runway, how much money you have left, with also, feel very bad, hire somebody who's amazing and say, we only have 12 months of money left. So, you might be out of a job in 12 months. So there's a lot of emotional things you have to consider.
Well, I think actually the fact that you are even thinking about that tells me that you're doing a better job than a lot of people do, because I've actually been not told that before. So
I know.
I actually think that you can find those people just by telling them that up front. Some people want to go on that journey with you, and part of it is you might run out of money in 12 months. You might also do something amazing and actually grow the company and be one of the first early people on board in an amazing company. And for people that are early in their career, that's what they want to do, right? Those are the risks you should take, especially early on in your career.
No, it's strange. I said when we started this company, my personality has definitely changed a little bit from being completely optimistic and everybody that's in the sciences is, has a very strong moral compass and always wants to do good, to sometimes you just have to accept there's a reality to the world. And I haven't become an asshole, and I still want to hold empathy and treat people well, because I think it works out in the long run than being short-sighted and greedy.
I think it's just experience, man. It's just wisdom that's come from age and experience. I think you're still a good person and want to do good in the world, but you also have to live within the confines of reality of the world around you. And that has, certain aspects to it that you have to consider. So how do you feel about your business? I mean, you feel like you're going to make it? Do you feel like, you have some...
Oh yeah, I still get up every day and I get excited. And especially when I do spend some time in the lab, I'm like, I remember why I'm doing this. And, I think we've made more progress in the year and a half since we switched to a new industry than prior. It's an amazing learning experience. And yeah, I'm highly optimistic and think things are going to work out.
Awesome, man.
Then I just want to do what, the people that gave us our first accelerator chip, I want to go back and invest in all these new people coming up with interesting scientific ideas. And
I can see that. I definitely think you will. I definitely think you've got like an amazing future ahead of you. You have like a lot of really exciting things in the works. And whether it's this company or the next one, every single time you go through these journeys, you pick up some new knowledge and experience and you take that with you. And if you just keep working like you've been doing, man, it's going to pay off. It definitely will. I'm definitely supporting and believing in you. You've been really helpful for me, by the way, behind the scenes. So
thanks.
I appreciate that. If someone wants to find you, like you do have a YouTube channel, I don't know if you want to share that, but if folks want to find you on YouTube or Instagram, or even if there's someone that maybe wants to come work for you, what would be the best way for folks to get in touch with you?
Yeah, you're welcome to find me on the Basement Biotech Instagram or YouTube. I don't know if giving my personal email is a good idea or a bad idea.
Give me like info or contact at or something.
No, I know. I mean, I think YouTube or Instagram. I have a lot of basement biotech stuff, but that started a long time ago. I've just kind of kept that hashtag because I like it. I
like it too. It's cool.
Thanks. I wish I don't make tons of videos anymore because this takes up so much of my time. But I'm super excited that you even reached out, and how we even met was so weird, looking for used glassware years ago. But I'm pumped that somebody like yourself is, one, getting into all this, but also doing what I wish somebody had done years ago, which is kind of add some more cohesion and coalescence to everybody's story, because it was mainly a bunch of random faces that you saw on a Google thread, and then all of a sudden you read, oh, that kid raised $2 million and he's making open PCR. That kid in Austria was doing something.
Well, man,
there's a lot of interesting people. There's no.
Yeah, absolutely. And I get so much energy just by talking to people like you who have way more experience in this stuff than me. Like, I would love nothing more than just to sit and talk to people about this stuff all day long, because I learn just by asking questions and seeing what other people are doing. So if you've got other people, by the way, that you think would be interested in doing this,
oh yeah,
definitely,
I can send you some notes after.
Yeah, please do. And I'll send you some updates on how I'm going to publish this, and try to make it professional, get it more and more professional, and hopefully maybe get it on iTunes if I can. I've got to figure that out. But
I don't think I've said anything too crazy out there. So
you haven't said anything.
Should be all good for me.
Can you give the URL to your company? You have a website, right?
Yeah, I mean, the company's Prospective Research. We put barely anything on there.
Yeah. But there's a contact information on there. That's what I'm getting at. Like if someone wanted to reach you.
Oh yeah, info at I think YouTube, my personal email, which is probably in there, will eventually get to me. Or find me on the DIYBio forum. My email's actually on there, so it's dcoats@gmail.com. So.
By the way, not everybody knows what that DIYBio forum is. If you Google DIYBio forum, it's actually a Google group, I believe. Right.
Yes, I always call it a forum, but it is a Google group.
I think if you just search for it, it's a Google group that's called like the DIYBio Google group, if I'm not mistaken. Yeah. Well, dude, thank you so much, man. This was awesome. I'm going to try to get this thing out the door tomorrow, hopefully, do the whole social media blast on it. And then I got one scheduled for the next 2 weeks, so I'm pumped about that. But thank you.
Nice.
Man. Your story is awesome. And I'm looking forward to following you and keeping in touch. And let me know if there's anything I can do to help you with everything going, man. I'm supporting you and rooting for you 100%, man.
Thanks, Josh. I'm supporting you too. I want to see lots of cool videos and famous iTunes podcasts from EverymanBio. Is that a Spore Print t-shirt? That thing's pretty cool.
Yeah. Nice. Thanks, brother.
Looks like Sauron's eye.
All right, man. Thank you, dude. All right, I'll see you.
Later.